It should work. Yes. Okay. So you can see this big screen here. Okay, perfect. So, before we dive in today, I just wanted to, start the workshop by talking about this concept that we talk about a lot in psychology, and we talk about a lot in psychopathology, and we talk about it a lot in development, which is this concept of developmental plasticity. So I wanted to start today by just defining this term plasticity a little bit more. So what is developmental plasticity? So when we talk about plasticity, or when I do, it's the quality really of being easily shaped or molded, kind of like Play-Doh. So we have this plasticity throughout our lives, and we can always be changed and molded to some extent by the environment. And that's usually what we call learning. So plasticity is always there throughout our lives. We can always learn no matter how old we are. But the term developmental plasticity really refers to this idea that particularly early in life, so infancy, childhood, adolescence even, we're particularly responsive to shaping and being molded by our environments. And this is importantly true in terms of behavior, but it's also importantly true at the level of the brain. So our neurons and our connections between them are very easily molded early in development. So in my lab at UCLA, the Brain and Body Lab, we study developmental plasticity. And we do this by asking about how the most important environment of childhood and adolescence, that is the home environment, and the caregivers who live within it, help to mold children's brains and bodies in ways that give rise to both mental and physical wellness, as well as mental and physical ill health or disease. And so in the lab, our mission is really to do good science that makes a difference to people's lives today and tomorrow. And so to do that, to achieve this aim of the lab, we engage in a variety of things. So one of the things that we do is disseminate knowledge about brain development, about early experiences and plasticity. And so that's the reason, one of the reasons that I'm here today is to tell you about what we do. So we run the research, we publish it, and then it kind of feels like that's the end of the process for many different labs. But what we're really trying to achieve is to bring it back to the people who participated in the research potentially, or whose lives the research may potentially impact. We also really like to engage the community in our research decisions and processes. Just going to admit some people. And that's another reason that I'm here today, is to engage you and to get feedback from you. So really, I am inviting you, if you have any feedback about the talk today, things that you're interested in, things that you think we're not researching that we should, I would just love to hear your feedback. So please don't hesitate to reach out. And then finally, we try to create meaningful knowledge through the research that we do in the lab. So although, I mentioned that this is basic research, so that means that we're not doing clinical trials or anything like that. We do try and do research that will actually potentially be moved into clinical trials, or potentially has a benefit to the communities who are engaging in our research process with us. So today, what I wanted to do in this workshop was to talk about 4 different areas. So firstly, I wanted to show you some of the ways that we as researchers look at children's emotional development, and what that tells us about the way that children grow up to be happy and healthy adults. And I'm going to describe studies to you that I've picked out that primarily show how phenomenal children are, honestly, how incredible the child-caregiver relationship is. And the reason that I've picked out these studies is really to empower you as a group of caregivers, as a group of professionals working in this space, to see the tremendous effects that you have over the children in your lives. And this is really, really ingrained in us as humans, down to the level of our biology. It's hardwired into us. And I think, being a caregiver, it's really important to understand and acknowledge how critical your role is as a parent, as a caregiver. And the other reason that I picked the studies that I'm going to show you today is because they tell us mechanistically why children can struggle so much when their relationship with their caregivers are disrupted early in life. And so it gives us some context to understand, well, now, when you see children potentially who haven't had caregivers around, or who may have had very disrupted or unstable caregiving experiences, why that tends to have such a big impact, and why it tends to have such a big impact in the domains that it does. So firstly, I'm going to talk about how caregivers shape emotional development, what can happen to emotional development after disrupted caregiving. I also then want to talk a little bit about resilience and the positive effect of positive early life experiences. So, leaving on a kind of upwards note. And then, as Dorothy alluded to earlier, I spend a lot of time looking at the connection between brains and bodies. So not just looking at the brain in our head and the psychological processes going on in that lump of tissue, but also looking at the rest of the body. So how do we integrate what our thoughts and feelings are with what's going on in terms of our physical health and symptoms? So I'm going to dive right in now by starting with that first question. So how do caregivers shape our emotional development? So to the casual observer, I think it's quite obvious that at least in mammals, parents play a really critical role in the development of their offspring, even after they have left the womb. Now, as mammals, we are actually born immature, and the biological term for that is altricial. That means that the care of the infant really falls solely in the hands of the parent, and sometimes in some cases, other adult caregivers, so maybe an extended family. Now, parents really do everything for children, as I'm sure you're all very aware of. So parents feed their infants, they maintain their body temperature, they soothe them. And even from day one, parents begin this process of teaching their infants to become independent. And so there's this lifelong process of kind of, looking after all of the needs of the child, but also teaching them to do it themselves. And in no case is the role of the parent greater than in human development, where parents care for their children for seriously long periods of time, sometimes well into their adulthood. I didn't actually leave my parents' house until I was, I think, 24, maybe even longer for some people. You may have some reluctant to leave 30-year-olds hanging around, but definitely more so than any other mammal, we are taking care of our babies for a long time. And humans then are also very involved grandparents. And so once we have our children leaving the home as adults, they not too soon afterwards have children of their own, and then we take on this role of grandparents. So parenting is a really enormous part of the human experience. So that, of course, brings up this question of what is the purpose of childhood? So we take care of children for so long, so we're parents for such a long time. Why are they being children for so long? Why are they being dependents for so long? What's the purpose of this extended childhood? So really, some of the theories in developmental psychology are that having a long childhood means that our brain can test out the environment that we find ourselves in. So rather than the brain just developing the exact same way for everyone, for every human, we can instead adapt and be very plastic or mold ourselves to the wide range of environments that we find ourselves in. So in other words, our brain figures out what works for our early environment, and then it commits to that, or sets the brain in stone, so to speak, the best way of functioning in that environment. So one of the questions that often comes up then is, well, if plasticity is good, if it means that we can adapt to the environments that we find ourselves in, why should we bother setting it in stone? Why don't we just stay really plastic forever and just continue to adapt to all of the different environments that we find ourselves in? And that can be particularly frustrating as parents, knowing that sometimes the earlier environments children were exposed to are not ideal, and we wish that they would continue to adapt to the subsequent safer environments that they're in. And the answer to that is really that there's a cost to high plasticity. And this is particularly true when we talk about the brain and we talk about learning. And so the cost to plasticity, or the trade-off, is efficiency. So our brains do need to eventually reduce plasticity, what I call this stage of exploration or testing out the environment, and ultimately move into a mode of exploitation, where it makes the circuits that it has formed more efficient. Now, I just want to point out that this does not mean that the brain cannot change later in life. It simply means that generally more effort is required to change a way of functioning that was set down early in our lives. So we really need to spend some time breaking down those bricks and dissolving the cement in order to get that plasticity back. And this is a really important point to take note of, because I know it can sometimes seem a little futile or disheartening to talk about plasticity in kind of yes or no terms. Either you have it or you don't. And so again, just to reiterate, plasticity is there throughout our lives. It's just much higher earlier in development. Okay, so now that we know a little bit about plasticity and what it means to have developmental plasticity in this long childhood, now we want to know what that plasticity is used for. So what are children exploring during these early phases of life, and how do we as parents assist them in that process? And so one of the things that children are learning about or exploring is their complex emotional lives. Children are dealing with a lot of big emotions, and importantly, they're learning how to cope with those emotions. And this is really critical for humans because we live in social networks. So understanding how to display a wide range of emotions and experience those emotions, as well as how to control them, is really critical for effectively functioning within a social network and taking advantage of social networks. And so this is where caregivers come in. So one of caregivers' primary jobs is to help children regulate so that they eventually become good self-regulators. So the regulation of emotions initially comes from parents from the outside, with the hope that ultimately this will then be handed on to the child or the adolescent themselves, and they can do more of that independently. Thanks, Genevieve. That's an interesting point. So Genevieve says, I appreciate this point about undoing the bricks and doing that intentional work. I'd heard the last 10 years that the brain has plasticity and indicated that somehow it should be more easy. Yeah, so yeah, absolutely. So the brain definitely waxes and wanes in terms of plasticity across the life. There will never be as much plasticity in the brain in adulthood as there is in childhood, but it is important to recognize that there is always some level of plasticity available. Okay, so how do parents regulate their children? We know it's not an easy thing to do. As young people can sometimes have really strong emotions about events that we as adults do not necessarily find distressing. So for example, this young man had a cereal bar in his left hand, but he wanted it in the right hand. A full tantrum ensued, and the parent was probably left wondering, like, what on earth? Why is this such a big issue for my child here? Now that I have my own child, can post many photographs of him being very alarmed and concerned about things that I just don't understand at all. So children really do have a lot of emotions, and they find them really difficult to control them. But luckily, Mother Nature has bestowed upon caregivers a true arsenal or a toolbox of resources to help them regulate the emotional output of their children. And so I'm going to talk about some of those resources today in the domains of stress regulation, how parents help children to regulate what's going on at the level of their brain, and also emotional regulation as well. So let's dive into this easiest or behavioral level of parents emotionally regulating their children. So children take cues from their parents, and parents can successfully upregulate and downregulate their children's behavioral expressions of fear, depending upon their own emotional output. And so a really nice experiment of this that I like to talk about is the visual cliff experiment. So originally, this visual cliff was designed to test an infant's depth perception. And the basic idea here is that you have a safe plexiglass floor that goes along the entire contraption. And then there is a cliff that the grade of which is made obvious by this checkered pattern. So the infants can perceive, oh, there's a drop off here, a big drop off, one that I can't crawl down. And they begin to, they stop at the edge of the visual cliff. But, all the babies were safe there. There is actually plexiglass, and they're very safe to crawl across the edge of the cliff. And so typically babies who are at crawling age will back away from the cliff. They understand that this is a dangerous thing to do. They won't dare to cross to the other side of the cliff. But this behavior does actually depend on their interaction with the parent. So you can see here that the mother of this child is standing on the other side of the cliff, beckoning their infant to cross. And what they found in this study and in many different studies is if the parent, if the mother is showing beckoning signs, like reassuring signs to the child to cross across the cliff that they know to be dangerous, then the infants will override their fear response and they will try out crossing to the other side of the cliff. But the reverse is also true. So if this parent was on the other side of the cliff saying like, no, don't do it, it's dangerous, be careful, and showing kind of a fear face, then infants will, definitely go with their initial instinct, which is to back away from the cliff. So just through emotions that are directed towards children, even if no words are expressed at all, parents can facilitate a child behavior that would otherwise not occur, and they can also prevent a behavior from occurring. So parents can be very powerful in terms of their presence and the emotions that they show on their own face. We also know that there's a lot of evidence showing that parents' regulation of their children extends beyond behavior and actually impacts the children's biology. So, for example, at the level of stress hormones. So one great way to look at the effect of parents on children's biology is to look at the end product of activation of the hypothalamic pituitary adrenal axis, that is cortisol, which is released from the adrenal cortex as an end product of activation of the stress response. And so by measuring cortisol, we can get an indication of how active this axis was in a child. So how do we cause, stress in the lab? How do we cause it in a way that's, nice and not damaging to children? So a stress that they can manage. And one of the most popular ways of doing this in the lab is through what we call a psychosocial evaluation stressor, or the Tria social stress test. And so you'll recognize that that's very similar to what I'm doing now, which is to give a talk in front of an audience. This is one of the most reliable ways in humans that we can activate the stress response system. And so in the lab, these young girls in this study were asked to give a talk on a topic they were not particularly familiar with in front of a panel of judges who were all wearing kind of white coats and looking very stern. And so we knew that this was going to activate their stress hormone levels. However, in this study, so the young girls were 7 to 12 years of age, they knew that this would evoke a stress response. That's been shown many times before. What they wanted to do in this study was to look at factors that helped stress hormone levels return to baseline more rapidly after this stressful event. And so basically what happened in this study was that girls were assigned to either have no contact with their parent immediately after the stressful event, or they were allowed to text message their parent. They were also able to speak to their parent on the phone, so just had auditory contact, or they were able to see their parent in person, so they had both visual and auditory contact. And so then they measured baseline levels of cortisol, so prior to the stressful event, and then during the stressful event, and then at several points after the stressful event, to see which of these modes of contact, if any, had helped these girls return to baseline. And so what you can see here is that about 20 minutes after the stressful event, this is cortisol, so the end product of this stress response, being measured in saliva. And you can see that for girls who had no contact with their parent, their stress hormone levels were still elevated after the stressful event. For girls who were able to instant message their parents, they also still had elevated cortisol levels. These are higher than this group visually, but these are error bars. They're not overlapping. They actually were not statistically different from one another. So either no contact with parents or text messaging was not effective in helping baseline levels come back down quickly. But for girls who were either able to have a phone conversation, so verbal contact with the parent, or full contact with the parent, so seeing them in person, they had a much more rapid return of cortisol levels to baseline. So just when we think that we might not be helping our adolescents, so 7 to 12 years is a difficult age for parents to feel like they're able to buffer or help their children's stress reactions, we can see from this study that at a most basic levels, for looking at stress hormones, parents are really quite effective, particularly when we're able to talk to our children directly or see them and comfort them in person. So this role for parents, being a buffer, does appear to be specific to childhood, as a study which also used that same social evaluative stressor in children versus adolescents. So children were 9 to 10 years old and adolescents were 15 to 16 years old. They did the same stress and tried to look and see whether parents had a different effect in children versus adolescents. And so this time in this study, they had children prepare for the stressor in the presence of their parent or in the presence of a friendly stranger. And so they wanted to see whether preparing for the stressful event with someone supportive nearby, so with the parent nearby, would help to reduce those stress hormone levels. And what they found in children is that children who were able to prepare for the stressful event in the presence of their parent, they actually didn't show that big increase in stress hormone levels after the evaluative stress. However, those who prepared in the presence of a stranger, they actually did show that increase in cortisol levels. And this is relative to day 2, which is just at rest. So these are their baseline levels. In adolescence, it looked a little different. So for both adolescents who had to prepare in the presence of a parent or stranger, everyone showed a stress response, which then returned to baseline. And there was no difference for the parent and stranger groups. So these were not statistically different than one another. So parents seem to be particularly effective during childhood to prevent this stressful experience from being kind of stressful at the level of stress hormones in the body, of cortisol. So we can actually see the amazing effect that parents have on children's biology, not only at the level of stress hormones, not only at the level of behavior, but also at the level of the brain. So this master controller that we have in our head that organizes everything that's going on in our body. So many of you may have heard about this brain region before, highlighted in red here. So this is a side view of the brain, and so I'm kind of taking a photograph from this direction. And this one is a front view of the brain, so taking a photograph right through the front. And this small almond-shaped structure in red here is called the amygdala. And the amygdala is a very popular brain region. It gets a lot of press. And one of the reasons it gets a lot of press is because it's involved in our emotional lives. So some people have described it as the fear center of the brain. That's a bit of a misnomer. It's not actually only involved in fear. It's actually strongly activated by fear, but many other different emotions. But it's very much a brain region that's important for our emotional lives. And in particular, it's very important in responding to highly salient or important emotional events. So interestingly enough, the amygdala is also very responsive to the presence of caregivers. So when we're looking at caregivers, when we're interacting with them, it tends to have a rather strong effect on what's going on at the level of the amygdala. So basically the way that we can look at the effect of caregivers over this brain structure is to have children lie in an MRI machine. So some of you might have had an MRI on different parts of your body before. When we do an MRI on the brain, we have children go in this direction, and they have their head in this, what's called the bore here, and as they go in, and their feet are down on this end. And basically an MRI is just a large magnet that helps us take photographs of the inside of our body. So children are they're certainly alive. Children are awake, I was going to say, in the magnet, and they're usually doing things like playing games or watching movies or looking at computer screens and making responses while they're in the MRI scanner. So in this study, they had children and teenagers lay in a scanner and simply look at photographs of their own parent or a stranger. So here's their own parent, and here's a stranger who was a race and age matched, other race, age and sex matched person from the study. So someone else's parent was acting as the stranger. And so we had the children lay in the bed and look at simply pictures of the parent and stranger. And so then they measured amygdala reactivities, so how much the amygdala responded to the parent relative to the stranger. So you always have to do a contrast like that in these MRI studies. And so what you're looking at on this particular graph here is the parent minus activity to the parent minus activity to the stranger. So any scores below 0 here mean that activity to the parent was lower than reactivity to the stranger. And anything above 0 is the opposite, so higher to the parent. And what's clear is that children in this study, so up to 10 years of age, actually showed amygdala buffering when they were looking at pictures of their parent. So in other words, their amygdala reactivity was decreased when looking at a parent relative to when looking at a stranger. For teens, they actually saw the opposite. So for teens, amygdala reactivity was slightly higher when looking at pictures of the parent than when looking at pictures of the stranger. So just by looking at their parent's face, this brain region, which really responds to and pays attention to these salient or strong emotional events, was dampened a little. So parental stimuli can help children to dampen or regulate their amygdala output. Now, there are also studies in rats that show that parents can buffer this brain region, this amygdala, and that this buffering, so parents' ability to dampen what's going on at the level of the amygdala, actually impacts what infants are able to learn. So in this study, what they did was simply to expose rats of different ages to two different odors. So they had a lemon odor here in yellow and a blueberry odor here in blue. So the lemon odor in this study was inconsequential, so they were exposed to it and nothing happened. And the other odor, blueberry, was consistently associated with a foot shock. So then after these animals learned this association, blueberry is dangerous, lemon is safe, then the researchers put the animal into a Y maze and they're allowed to just freely explore. And so the measure here is, do the animals want to spend more time in the safe zone with the lemon odor, or do they want to spend time in the dangerous zone with the blueberry? And so what you see in grown-up rats, so this older age group here, is no surprise. They avoid the arm that was paired with the foot shock. So they go straight to the lemon odor. And this behavior is accompanied by a really strong activation of the amygdala. So when you measure the amygdala in these animals, it's really firing. So these animals were able to learn blueberry is dangerous, amygdala is on, it's paying attention, and they're able to avoid that dangerous scent and go towards safety. Now, in these young animals, they do the same thing. So their amygdala is on and they avoid the dangerous odor. However, if the young rat learned while the mother was present, so if you have the mother there while the animal is learning that lemon is safe and blueberry is dangerous, their amygdala is actually suppressed, turned off, and they actually don't learn to avoid that dangerous side of the maze. So they don't learn that blueberry is associated with foot shock and to avoid it. So just by having the parent present, it can so drastically dampen amygdala reactivity that it causes animals to paradoxically, weirdly go towards this odor that they know to be dangerous. So in other words, just through their presence, mother actually can switch on and off the amygdala to alter infants' behavior. And so when we think about that, I'm always just astounded by these studies because it's almost like a pharmacological effect. Parents are so powerful here that they can literally switch on and off this particular brain region. So just to summarize what I've shown you so far, caregivers are really important at regulating children's emotional output. And they can do this behaviorally. They can do it at the level of stress hormones, and they can do it by engaging or reducing engagement rather of brain regions involved in emotional reactivity. So I've spoken a lot about the amygdala here, but there's lots of research looking at the medial prefrontal cortex and other brain regions like the hippocampus. So parents are really important at multiple different levels of children's biology and psychology to alter behaviors, and particularly emotional behaviors. Now these, and actually in addition, there seems to be a special period for these effects in childhood that seems to dampen or wane as children go through adolescence. Now, these effects, I think, really make sense when we consider our evolutionary history. So that is, there's a species expectation for mammals, so whether you're humans, monkeys, or any other sort of mammal, that we grow up with caregivers around. It's one of the defining biological features of being a mammal. So having these caregivers around really affords us this long period of childhood, this long period of maturation, where we know that our caregivers are looking after us and we can invest our resources in our brain to learning about the environment, and particularly learning about this rich, complex social and emotional life that we're going to need to inhabit after we leave the nest, after we leave home. And of course, that makes us think then, well, we know that while this is what the species expectation is, so across all different mammals, the species expectation is that the caregiver will be there. It, of course, makes us think, well, what happens when caregiving goes awry? So what happens when that species expectation is not met? So in the next part of the talk, I'm going to talk about that more. So disruption of the caregiving relationship and what the consequences are for emotional development. But just for the moment, I wanted to stop and quickly see whether there are any questions or comments that we had for this first part of the talk. Any clarifying or larger questions, I'm happy to answer. Okay, that's great. So, and again, anytime you have any questions, please don't hesitate to stop me. Okay. So now I'm going to talk a little bit about some research that we've done looking at brain changes when that expectation of parental presence is not met. Oh, thank you. Thanks, Laura. So you've said, is there any difference in hormonal and brain engagement in biological caregivers? Relative to what, Laura?
I guess I just wanted to know, does it come from a biological connection, or is it just somebody that's been there?
It's a really, really good question. I think this next part of the talk will help to answer that. But in short, no, it's not a genetic connection. It's a presence effect. And in particular, it's a presence during a particular stage of development effect. Thank you for that question. And anyone else, if you're happy to talk, then just interrupt me or throw it in the chat, and I'm trying to monitor the chat as we go along. Okay, so as I said, we're going to talk about some research that we've done looking at brain changes when that expectation of parental presence is not met and what the consequences are for children's emotional development. And so here I'm going to kind of go in reverse and talk a little bit about the non-human animal data first, as I think it's particularly informative in terms of helping us understand causality. And I think that's something that's very hard to understand in humans, but I will get to some human data immediately afterwards. Okay, so picking back up on that last study that I showed you in rats, we saw that the mother was able to block amygdala reactivity when the young rat was exposed to a stressor, and they found that blocking amygdala reactivity actually caused these animals to go towards the scary or dangerous side of this Y maze. However, in this study, another thing that they did was to expose these pups to disrupted caregiving early in life. So in particular, what they did in this study was to have mums in a cage with their pups with very limited nesting materials. And so rat mums are very good at building these wonderful, big, safe, cozy nests. But if they're not given enough nesting material, then they spend all of their time panicking, walking around the nest, or rather around the cage, trying to build a good nest and not succeeding. And in the process, they can be very harsh parents to their pups because they keep picking them up and stepping on them and frantically moving them around, trying to get them to safety. So it's really a model of low resources and consequent harsh parenting. And so they find that when animals are exposed to this very different type of early environment, then the mother was no longer able to block amygdala reactivity in the pup, and that pup would start showing fear reactions. So this pup actually now learns, hang on, this is actually dangerous even though my mum's around, and I'm going to start avoid that blueberry odor and go towards the safe side. So in other words, young pups that had exposure to disrupted caregiving actually started to behave like adult animals because the mother could no longer modulate their amygdala. And so they lost that developmental plasticity. And so there's a suggestion here that maybe their development in terms of their fear learning and their emotional development was somewhat accelerated following adversity. So because the mother wasn't really around to help protect them, or she was, occupied with other things, then these animals kind of grew up more quickly and they began to behave like adults. So they, regardless of maternal presence, always learned about the danger in the environment and subsequently avoided it. So there's another example, actually there's many in the literature, but there's another one that I'm going to talk about today of accelerated development after caregiving adversity in rats. And so in this study, rats were exposed to either standard rearing, where they remain in the home cage with the mother from birth, or maternal separation, where they're separated from the mother for a few hours each day. And this is actually abnormal for rats, even within the wild. So typically the mum will only leave the nest for periods of about 20 minutes to forage for food. She would rarely be gone for several hours at a time. And so this is a model of deprivation, so where caregivers are not around or neglect. And then when these animals, these young pups reach childhood, they're trained to associate a noise with a foot shock. And they're then tested for their fear reactions to that noise, so this predictive cue, either immediately after they've learned or at a delay. And so what typically happens, so across in the standard reared animals at this age in childhood, is that they rapidly learn and express fear towards that noise. However, they rapidly forget that fear association. So in other words, when pups are around a parent, so the caregiver is present a lot of the time or all of the time, then they do learn fear associations, but they rapidly forget them as well. So there's an idea here that they can learn about things that may be immediately relevant, but because their parent is around, they ultimately don't need to hold on to those fear associations for long periods of time. In contrast, for the pups who were exposed to this deprivation or neglect, maternal separation, they actually behave more like adults. So they show really strong fear responses, regardless of when they're tested, either immediately or at a delay. So again, this seems like development might be accelerated following a period of neglect or adversity in the pups. So Laura's saying, so close caregiver kind of provide a bubble of safety for the one they're caring for, and they have less fear. Exactly. That's exactly right, Laura. Yeah, so it's that having the parent around does seem to show the young rat that they're in a safe space. And so they don't need to fear themselves. They can learn about things, but they don't need to hold on to those associations for long periods of time. However, if parents are not consistently present, then those animals do need to start learning and fending for themselves. So do we see any evidence of this accelerated development, this kind of fending for yourself behavior in humans? In fact, we do. So going back to this earlier study where we had children laying in the scanner looking at pictures of their parent or a stranger, this study was done in kids who had biological or birth first parents. On the left here, so this is the response of those children. And we also did this study in children who'd been adopted, and in this case, children who'd been adopted from early institutional care into American families. So institutional care abroad into American families, so internationally adopted kids. And so in this study, over this side, these children were looking at pictures of their adoptive parent. And so, kind of getting to Laura's question, these kids had been with these adoptive caregivers for a really long period of time, but everyone was adopted by about the age of 2. So everyone had been with their adoptive families for long periods of time. And what we found here is that overwhelmingly, so across at the group level, the children did not show this amygdala buffering effect. So children who'd been exposed to this form of early caregiving adversity didn't show buffering to pictures of their adoptive parent. But getting back to Laura's question, what you can see here is that there's huge variability in this group. So for those of you who may be less familiar with these types of graphs, what you're seeing in the bars is the group effect. So this is the mean. But these lines that go over the top, these are what we call error bars. And this indicates how much variability there is in the data. And so you'll see, particularly within this child age group, for those who'd experienced early caregiving adversity, there's lots of differences. So clearly, although at the group level, children who'd experienced early caregiving adversity were not showing amygdala buffering to pictures of their adoptive parent, that wasn't true for everyone. Certainly some of these children were. And so we wanted to dive in and see, well, what does this variability relate to? Is it just noise, or is there something meaningful behind it? And so what we did was to look at anxiety as a function of whether children exhibited buffering at time 1 or not. So we had data at the time of the scan, which was time 1, as well as 2 years later, so year 3 of the study. And so what you'll see within the comparison group on the left here is that buffering really didn't seem to matter. Anxiety levels were always really low in children who had birth first or biological parents. On the right, you'll see that overwhelmingly children who'd experienced early caregiving adversity did have higher levels of anxiety, but your anxiety change over time differed according to whether you were a bufferer or not. So specifically, those children who were able to show amygdala buffering to pictures of their caregivers at time 1 showed a sharper decline in anxiety into the future, into year 3 of the study. So this, of course, makes us ask, well, what is it that is associated with buffering in this group? Like, why are some children exhibiting buffering and some children are not? Especially considering we know that this buffering isn't just noise. This variability in buffering isn't noise. It seems to be related to anxiety and real-world outcomes. And so what we did is actually look at parenting and the security in the parent-child relationship as reported by the child. And so this graph is just within children who'd been exposed to early caregiving adversity and international adoption. And we split the data into those children who exhibited parental buffering at time 1, so on the right, and those children who did not. And it was pretty clear that those children who were able to exhibit parental buffering to their pictures of their adoptive parent at the level of the amygdala, they were reporting high feelings of attachment security in that relationship with their adoptive caregiver. So although there's this group effect where children exposed to adversity seem to be different than children that were not exposed to adversity at this basic brain level, so the response of the brain to pictures of parental cues, that really does depend on the quality of their parent-child relationship and how secure children are feeling in that relationship. In other words, those post-adoptive variables really made a huge difference. So to summarize this part of the talk then, adversity seems to accelerate emotional development and associated neurobiology in rats as well as in humans. And this is, a limited window here that we're looking at these effects, but we do see it across many different studies, particularly looking at this system of amygdala and threat and fear learning. But as I've mentioned, there is tremendous variability in the outcomes, and these appear to be related to long-term mental health. And in particular, strong child caregiver relationships are associated with more positive mental health outcomes for youth, particularly youth who've been adopted or who've experienced caregiving adversity in early life. And so again, these effects really make sense when we consider our evolutionary history. So again, there's this species expectation that mammals, humans, and other animals grow up with the caregivers around. This affords us this longer period of maturation, but that's a luxury. When caregivers aren't around or when caregivers are not reliable, we do need to grow up faster and our brains and bodies react differently. And we get into a position where we're more self-reliant. And there might be consequences, or there do seem to be consequences of that developmental pathway, that trajectory for our emotions. So next, I want to continue making this point about the impact of positive early experiences. So, we've been talking about these post-adoptive factors. So what are the positive experiences that children have in their life, and how do those operate in the context of adversity? So what are positive experiences and how do they work, and do they matter when our children have been exposed to significant challenges or adversities early in life? So in this study, this is actually a semi-recent study now, so done in 2019. And I really love it because they've asked this really interesting question of resilience or buffering factors, both inside and outside of the home, in the context of really high ACEs, in the context of high adverse childhood experiences. And so what they've done in this study was to firstly look at depression rates in adults who had high adverse childhood experiences, or ACEs. And they also measured positive childhood experiences in those adults. And what they saw is that depression levels were higher in adults who had high ACEs than in the general population. That's been shown many times before. But they did see that positive events really made a difference, even within these high ACEs individuals. So individuals who had high ACEs, but also very low numbers of positive events, they actually had the highest rates of depression. But if individuals had high ACEs, but also had many positive events in their early life, then they actually showed depression levels that were much lower, actually nearing the levels that we see within the general population. So this is really exciting because it means that positive events can interact with our early life experiences to put us on different developmental trajectories. Got a chat question here. That is so hopeful, Genevieve. I really agree with you. I loved reading this study. And then it made me think, well, what are these positive early childhood experiences? They must be truly amazing, right? If we're to overcome all of these ACEs, if we're to overcome all of this early life adversity. And it turns out they weren't really that amazing. They were these everyday things that children can experience. There was not some kind of magic feather here. So some of the examples were being able to talk to your family about your feelings, feeling like your family stood by you during a difficult time, feeling safe and protected by an adult in the home, having at least 2 non-parent adults who took a genuine interest in your development, feeling supported by friends, feeling a sense of belonging at high school, and enjoying participation in community traditions. And this is not an exhaustive list. These are just some that I've picked out. So you can see here that ACEs are not deterministic in life outcomes. Positive experiences, and particularly relationships with other caregivers and adults, these make a huge difference in terms of mental health outcomes later in life. So we've spoken a lot about typical development or development, without a significant caregiving adversity. We've talked about development in the context of high caregiving adversities, which paints a very different picture, but we've also spoken a lot about these resilience factors. So the post-adopted factors, as well as that study with all of the different positive events that can happen in one's life. And, in the process of doing that, I've spoken a lot about the brain, I've spoken a lot about behavior, and I've spoken a lot about mental health. And that's okay because I'm a psychologist. So, talking about mental health really is my wheelhouse. But when it comes to early life experiences, we know that they're not just important for mental health. They're also really critically important for our physical health. In other words, there's this really powerful connection that takes place between our brains and our bodies. So I wanted to spend this last section of the talk today discussing a little bit about what we know about this brain and body connection in development and how disruptions to caregiving can affect that connection as well. I do want to just pause very briefly and see whether there are any questions at this stage. No? Okay. No worries, just dive in if anything. All right, so let's talk about the brain-body connection. So there's certainly a lot of evidence showing that physical health is connected to early adversity exposure. So in this particular paper, so Hughes in 2017, they looked at 37 different studies that had measured adverse childhood experiences and that looked at a physical health outcome. And what they found amongst those 37 studies is that adults with high ACEs had increased risks of experiencing all of the physical health disorders or diseases that were measured, including things like heart disease, obesity, and drug abuse or use. So seeing statistics like these has made me personally very interested in understanding how early life experiences shape not only our mental health, but also our physical health. And even more important than that, how our mental and physical health interact or work together or separately to set individuals on trajectories of health and disease. So how do physical and mental health interact? Why are we seeing that adverse childhood experiences are so important for our whole body? So in my work, I've been focusing on a physical system that we know is very tightly connected to mental health, and that is the gastrointestinal system. So we know in adults that functional gastrointestinal disorders like irritable bowel syndrome or IBS are really strongly associated with the incidence of childhood trauma. So for example, in adults, we know that adult women with IBS report a higher prevalence of general childhood trauma than control women. This is one study of many that has reported this finding. And it's not to say that all people with IBS have childhood trauma, but just that there's a higher representation of childhood trauma within that group. We also have a lot of anecdotal evidence in our language as adults that tie gastrointestinal symptoms to emotional experiences of anxiety and fear. So I'm sure we can all attest to, having spoken about having butterflies in our stomach, or having our stomach dropped in fear, or having a gut reaction to something. And I'm sure a lot of you can identify with that kind of gastrointestinal feeling of anxiety and fear kind of being situated in the gut. From animal models, we know that early separation from the caregiver, so that maternal separation in rats, is associated with irritable bowel syndrome-like symptoms in early development. And we also know that anxiety and gastrointestinal complaints are actually very common together. They're comorbid in children. And in fact, gastrointestinal complaints that are unexplained by a clear medical cause, that is, they don't have an organic origin, are very, very common presentations to primary healthcare facilities. So lots of children have these unexplained tummy aches. And the thinking is that they may be really tightly associated with mental health there too. In my research, we've now shown across thousands of children in different populations, shown in different colors here. So there are 3 different cohorts totaling, I think over 2,500 children, that there's a very strong relationship between anxiety and gastrointestinal symptoms. So high gastrointestinal symptoms is associated with higher anxiety. It's a really consistent finding. And so we've seen it, as I mentioned, in my lab several times, but many different people have reported this association. And so the question here is, well, what's going on? Why are gastrointestinal complaints so common in anxiety and vice versa? What are the mechanisms underlying this association? And so we now know that the brain, the gut, and the thriving mass of bacteria that exist inside the gut, that is the gastrointestinal microbiome or microbiota, participate in a bidirectional communication highway called the brain-gut microbiome axis, or sometimes the gut-brain microbiome axis. depending on who you are, it depends on what gets to go first. Now, communication along this highway is incredibly complicated, and I'm showing you this very pretty but super complicated graph, not to kind of put you off, but just to illustrate that exact point. This is a super complicated system. So we've got another little chat. Okay, I'm going to leave that one until later on. But thank you, Laura. This is great fodder for the discussion section. So now communication along this highway is incredibly complicated. There's a role for stress hormones. So for example, cortisol, I was talking about cortisol earlier. There's also a role for immune cells and our immune functioning, our vagus nerve. Bacteria produce neurotransmitters in the gut, and that then influences the brain. And it's most importantly bidirectional. So there's things that are happening at the level of the brain that can influence what's going on in the gut, and vice versa. Things that are happening in the gut ultimately end up feeding back to the brain. Now, one thing to point out here, as I already have, is that stress hormones, and particularly cortisol, play a really critical role in regulating communication across this highway. Now, indeed, the very process of brain development itself appears to be, at least in a small part, dependent on the microbiome. So here is a depiction of a rat brain. It looks very different than a human brain. The eyes are down this end. And here I've just got a very crude picture of different brain regions. So one that I'm going to point out here is the amygdala. So rats have an amygdala as well as humans. Most animals do. And the amygdala, so its development is really dependent on there being bacteria in the body early in life. So if the rat grows up germ-free, so in an isolator with no other bacteria, no bacteria around, then the amygdala develops very unusually. So there's increased volume of the amygdala, the dendrites actually shorten, the dendritic spines shorten, and there's abnormal levels of growth hormone in this structure. Similarly, with the prefrontal cortex, a very critical brain region for our emotional health, there's effects there, as well as with the hippocampus. So not to kind of get into the weeds, but just to make the point that without bacteria on board early in life, these brain regions really end up going a bit wackadoodle. So they really need that bacteria early in life in order to develop along the trajectory that we're most familiar with. And so that I find that really mind-blowing, that we actually have evolved in the context of these bacteria, and we need them to produce the brain that we know and love, that we're familiar with today. And importantly, these bacteria appear to have a programming effect on the development of these brain regions, because if you reconstitute the microbiome, so if you give bacteria after they emerge from the bubble, let's say after infancy or childhood, that actually doesn't help to reverse many of the brain patterns that we see here. It helps to reverse some of them, but not all of them. So in other words, we need a microbiome in order to build a brain. Now, because the gut seems to be so strongly associated with brain development, and in particular with development of brain regions that are associated with our emotional lives, and because the brain-gut microbiome axis is affected by stress hormones, there's a lot of smoking guns here. So there seems to be a lot of clinical promise in the microbiome for emotional health. For example, one day we hope to be able to target the gut to affect brain function and behavior in humans. That's something that, you know, the scientific world is talking a lot about at the moment. And maybe using gut treatments to enhance therapies that are already commonly used for mental health. So for example, cognitive behavioral therapy. And there are at least 2 studies now that are in the early stages, but 2 published works that are showing that that's actually feasible, that may actually work. So these are questions, and they're open questions. We're gaining evidence on them. But the only way that we're going to be able to answer them is to learn a lot more about the microbiome, its development, and its connection to the brain and mental health. So recently, my lab has been trying to understand the process of brain development and its relationship to bacteria in humans by studying the microbiome. And one of the ways that we do that is through collecting poop. So we fondly call this study the Poop Fairy study, and I'm going to describe for you the pilot phase of the study and show you how we're collecting more data in my lab to replicate these results in a larger sample and to really extend the findings. So in our Poop Fairy study, what we did was to call up a group of individuals who'd already taken part in some of the research in the lab and simply ask them if they wanted to donate a stool sample. Now, many of you will not be surprised that children seem to be largely very happy to donate a stool sample. Toilet humor, poop doesn't seem to affect children. They find it quite funny and interesting. So we got lots of children interested in donating a poop sample from home. So what they did was do their business in their safe and private confines of their own home, and then they popped a tiny little chunk of poop into a tube and mailed it back to us in the lab. And then we sent the Poop Fairy over to their house, and that Poop Fairy gave them $25 for their stool. So very generous fairy here, much more generous than the Tooth Fairy. And then we did what's called gene sequencing on the bacteria to ask who lives in the gut. So who is a taxonomic question. I'm just trying to keep up with the chat. Something while I'm medical, just curious. Sounds like it's still being studied. Can you say more, Genevieve? Great.
Trying to unmute. I probably shouldn't have said anything because I don't want to be disruptive, but I had just read some stuff on gut health.
Yeah.
That I can't remember because it was like jotting it down, trying to learn. I had heard it before, but I was trying to like really apply it now. As this is all tied to global warming and everything, we're trying to get more healthy here and eat less meat. Are you still there? Okay, a phone call had interrupted, sorry. So I was just curious if it was like basic stuff like getting more live cultures from yogurts in our diet for our kids, or if it's like something more medicinal that you're referring to. I was just trying to wrap my head around what you were saying.
It's a great question, and it's one I get a lot. So the easy answer is that we don't know yet. So it might be something really easy. Maybe it's just we need to eat more green foods and feed the good bacteria in our gut. Maybe we need prebiotics, maybe we need probiotics, or maybe it's something more targeted. And so I think a lot of the thinking at the moment, particularly within the microbiome world, so people who are actual microbiologists, is the exciting bit is that it could potentially be targeted. So can we use these bacteria? There are even ways to engineer bacteria in order to influence what's going on at the gut, the types of hormones that are produced in the gut, the types of neurotransmitters that are produced in the gut, and then non-invasively influence what's happening at the level of the brain, what's happening with behavior. But the short answer is that we are at such an early stage of this research, particularly in humans. You know, there's lots of preclinical work in animals going on, but we're at such an early stage in humans that we're just really needing to learn more. And then I'd even further amplify that and say in development, we need to learn a lot more. Yeah. But yeah, your thinking is along the right tracks. Is it bacteria? Is it probiotics? Is it, whatnot? We still don't know. So in this particular study, we were simply asking who lives in the gut. You can ask many different questions like what do they do, but we were asking a very simple question here, who lives in the gut? And so in this study it's a small sample size, as I mentioned, it's a pilot. There were only 16 kids and they were 7 to 13 years of age in this particular study. Half of them had been internationally adopted from institutional care, all before the age of 2 years. And everyone was now aged 7 to 13 years. So for the kids who'd been adopted, they had been living in the United States for at least 5 years by the time we were looking at their stool samples. So there are, as I'm sure you can imagine, many different ways of analyzing bacteria in the gut, and some of them get very complicated. So what I wanted to do today is just to show you something very simple, which is called diversity. So basically this question is, how many different bacterial groups are represented in the guts of individuals as a function of their early life caregiving environment. And so what we found is that children who'd been exposed to early caregiving adversity had fewer numbers of bacterial groups. So they've got 3 different bacterial groups here. I mean, it's just depicting it was more than that, but to show the point, fewer bacterial groups than children in the comparison sample. And so these were kids who were with their biological birth or first families. And so what we call this is reduced diversity. So we've reduced within person diversity of gut bacteria. And importantly, this was the first time we had looked at this. This is the first time anyone had looked at bacteria as a function of caregiving environments. But importantly, as I mentioned, these kids had been living in the United States for a really long time by the stage at which we assess their microbiota. So the opportunity for the Western diet, for example, and different things associated with being adopted to influence the gut had plenty of time to take effect, but we were still seeing these effects here. So I think that's an important thing to point out. Now, the real thing that we wanted to do in this study was to look at how bacteria were associated with the brain. And in particular, we wanted to ask a pretty simple question, which is, can we detect any associations between bacteria and the brain at all? So is this even a feasible thing to do? And so, as you can imagine, there are many different bacteria in the gut. Here are just the top 30 that we saw among children who've been exposed to early adversity and in the comparison group. What bacteria to choose in terms of looking at the association with brain. And so we ran an analysis that basically just uses machine learning to pick out a bacteria that most predicts or is associated with group membership. So this bacteria, Lachnospiraceae, came out as being most differentiating between the 2 groups. And in particular, it was higher in children who'd been with their birth first or biological parents always, and it was generally a lot lower in children who've been exposed to early life adversity, though we can see some variability here. And so taking that bacteria, Lachnospiraceae, so what we did is take the levels of that bacteria for each individual child, and we just ran a correlation with the brain. And so this is the brain while it's doing an emotion task. So this is the brain while children are looking at threatening faces on a computer screen. And so we know that type of task, looking at scary faces or threatening faces, like that type of task really strongly activates emotion circuitry, including the prefrontal cortex and amygdala. And so we were kind of biasing activation in those brain regions and saying, do individual levels of this bacteria, are they correlated with how much the brain activates in those environments, so those threatening places? And very surprisingly, we found that they were. So having this level of bacteria in your body, like Nospraceae at high levels, was associated with more activation of the medial prefrontal cortex and left lateral prefrontal cortex, and also some cerebellum here as well. And it was negatively associated with reactivity in the postcentral gyrus. So those are a lot of words that you may not have heard of before. We've talked a little bit about MPFC, the medial prefrontal cortex. You might not have heard about these other brain regions. Doesn't matter. The point is just to say that we do see associations between single bacteria that exist in the body and what the brain is doing in emotion tasks. So that's really exciting for us, but there's a lot more that needs to be done on this front. And in particular, moving beyond associations, we need to see that this replicates in many different people. And more importantly, we need to find out what this means. So we need to move bacteria around, look at it more in terms of a community context. And so all of that means that we need more people, more children to participate in this research. And so, we have a sample that's growing, but we continue to need more families who are interested in learning about these processes with us. So, I wanted to let you know that we do have a virtual research opportunity open at the moment. And if any of you are interested, or you may be working with families who could be interested, then please, get in touch and let us know. And so for this particular study, we're looking at children who are who've been adopted or who are in guardianship care, who are between 6 and 16 years old. And basically it's a Zoom session. So one of our friendly research staff logs on to the session with your family. You do some questionnaires, you play some computer games, and then we send you a kit so that you can collect some saliva, some stool, and some hair. And you get paid for your participation. So you can get from $45 to $65 for your participation in the study. Because it's virtual, you don't need to be in the UCLA area in order to do this. And we find it's a really fun way for parents and children to learn about social sciences together. So it's a good conversation starter for future careers in science. Maybe they're inspired to be a poop fairy themselves, or maybe not. It's all learning to be had. So just to summarize this section of the talk, what I've shown you is that caregivers really have a huge amount of power to positively affect their children's mental and their physical health. Children respond to caregivers at the level of their brain, their biology, and their behavior. And our brains and bodies are connected. So paying attention to both mental and physical symptoms is a sign of wellness. And I think that's a really important point that we miss a lot, as parents and certainly as healthcare providers, is that physical health matters just as much as mental health when we're talking about the overall well-being of a child. And so we really need to pay attention to both. And importantly, we might be able to use this knowledge to improve our existing treatments for mental health, as well as develop new treatments. And I'm going to use a word holistic, which I think generally tends to have a bit of a maybe a bad rap, or at least a bit of a woo-woo rap. But I think it's a really important word because it really gets at what we're trying to do here. So taking account of the whole child, all of their environmental context, their history, their developmental history, as well as their brain and body and how those things are interacting. And by taking that kind of complicated and holistic approach, then that hopefully is going to get us closer towards creating treatments that are non-invasive, that are things that parents and children want to do, and that may actually be very effective in helping the development get on track. So I'm going to end. I wanted to just point out that we have lots of resources in the lab. As I mentioned at the start, one of the things that we like to do is to kind of create meaningful connections with community. We've got lots of further reading. There's ways to participate in the studies, as well as join our community. So even if you're not interested in participating, you can join our mailing list. You can hear about the studies that we're doing and keep up to date with the research. Sometimes we have colouring in competitions for children and drawing competitions and lots of fun things like that to keep you all kind of involved as a community. So I'm going to end and say a really enormous thank you to my lab. The faces change all the time as students are growing and developing and moving on to their next stages of career. So this actually is not what the lab looks like at the moment, but these are some lovely people who've participated in the studies in the past. And to all of you, I really appreciate you taking the time, and I'm so excited now to talk about some questions.